Hello, and you're welcome back to the channel. It's always beautiful having you here, and we continue our discourse. Today, we are going to discuss about creating a continuous microbial culture. Now, what's a continuous microbial culture? There are instances where you want to, generate microorganisms. You want microorganisms to grow, and you want to have a continuous source of these microorganisms over a long period of time. You want to be able to harvest pure cultures of microorganisms over a long period of time. And now, this is important for many, many reasons. Now, the difference between a continuous culture and what we refer to as a batch culture is that in a batch culture, you have these microorganisms growing, and once they grow, the growth sort of comes to an end and then it ends there. But you can compare that with a continuous culture. A continuous culture is a culture in which you have this continued growth of the microorganisms. Now, there are many kinds of equipments that we can use to achieve this sort of thing. We have what we call a chemostat, and what we have another equipment we call a turbidostat. Now let's start with a chemostat. A chemostat is a kind of system where we have what we refer to as a closed system. Now a closed system in which you have microorganisms growing in a particular culture medium. Many a times it's broth culture, that's liquid culture. And then the system is maintained. Is maintained in such a way that there is the continuous addition of medium and nutrients through one end of the chemostat, and at the other end there's a continuous harvesting of the pure cultures, pure isolates of the organism over a sustained period of time. Now this is important and is used for many purposes. Now in a chemostat, it's important to realize that as the medium is being added, the nutrients that the organisms need to grow, very rapidly is also being added along with the medium. And so the organisms continue to grow in this medium. Now, in order to control this growth so that the growth doesn't get too wild, there's a control of the concentration of what we call a limiting factor. A particular nutrient, for an example, nitrogen, can be limiting in the sense that the growth of many of these microorganisms are tied to the presence of this particular nutrient. And so, in order to control the growth of these microorganisms, you can control the concentration of many of these, the concentration of this nutrient. So, this is done while using a chemostat, and this is really important to note. So, the chemostat is there and it can help us to do many things. So, just as I said before, you have the fresh culture medium, and then in the culture medium, you continue to add the medium and you continue to add nutrients, and then you harvest your pure cultures. And this is important to note. There's also something called a dilution rate, which is what we refer to as the rate of medium flow, and is divided by the rate of the culture volume. Now, this is important because it determines the growth of the culture and the things that we have in the culture. Now, what are some of the key features of the chemostat? The chemostat provides a steady flow of microorganisms. You have a steady flow of microorganisms. You have a controlled environment, and then you can use it to study microbial physiology. I use it to study a lot of microorganisms over a long period of time. Now we also have what we call the turbidostat, which also creates a closed system just like our chemostat. And also it's important to know that the chemostat can also be called a bioreflector. Now we also have the turbidostat, which is like the chemostat, and it's just a system also that uses turbidometry. Turbidometry, simple English, there's nothing difficult there. When you're talking turbidometry, you're talking about how turbid is a liquid. The more turbid it is, you know that the more there's microbial growth within that system. And the less turbid it is, the clearer the medium is, you know that there is less growth. So you can also have this kind of system being used or being run. That's continuous culture system can also be run with using a turbidostat. And if this is done, you still have the production of many different types of organisms. You also have what we call an optical density sensor, because we know anytime we are dealing with turbidometry, we're looking at optical density, how dense or not the solution is. That would tell you how much microbial growth you have or how little microbial growth you have occurring within that system. So all these things are important. Now, what are the applications of some of all these things? Some of the applications that they help us to be able to study microbial physiology and microbial kinetics. It also helps us in large industrial processes when we want to study the way microorganisms behave in large quantities. We want to study conditions that favor their growth. We want to study different kinds of aspects of microbiology, and we need large amounts of these microorganisms. We can get such by running a continuous culture. We may also want to do other experimental studies and ecological studies. We can do many of these if we have a continuous culture in place. Also, when we want to study antimicrobial resistance, do different kinds of resistance work, do check MIC, that's minimum inhibitory concentration, check MBC, check all these, even you are doing your LD50, all these different kinds of analysis that we can do. We can run them if we have a continuous culture. You think of a continuous culture as a factory where you are consistently producing microorganisms, which you can now use for many other different things that you want to use. Even if you are studying wastewater microbiology, you can also do a lot of all these features. So this brings us to the end of this our class. We've spent our time discussing what a continuous culture is, the importance of a continuous culture, two different types of equipments that can be used to create this culture, your chemostat, which is a bioreactor, and your turbidostat, which you use optical density to measure the rate at which microorganisms are growing within this system. Thank you for your continued following of these classes. Remember to like, subscribe, and share, and do have a blissful day.